Hello everyone, my name is Paul Limpin and today I'll be discussing bacterial meningitis and its causative agent in Neisseria meningitidis. This bacteria is also known as meningitis and is a treatable, preventable, but still highly virulent strand. A quick disclaimer I will make in this presentation is that I will use the terms Neisseria in reference to the bacteria and meningitis and meningococcus interchangeably throughout this PowerPoint, but the meanings of this virus will all still mean the same regardless of the meaning I use. So what is Neisseria meningitidis? It is a gram-negative diplococci species hailing from the genus of Neisseria. You may have heard of it through another member of the same genus through the name of Neisseria gonorrhoeae, but outside of their names, it's hard to tell which Neisseria you have if you were given an unknown member in a lab. So what's the similarities between them? So they are both gram-negative diplococcus. They are both catalase and oxidase positive. They're also aerobic anaerobes. They're both chemoheterotrophs, they have no flagella, and they share 90% of their genome between each other. So how do we tell the difference? Well, meningitis has the key difference is that it ferments glucose and maltose, which is not the case for gonorrhea. The other main difference is their pilus or pili. Meningitis has 4 retractable pilus, while gonorrhea only has 1. Meningitis also has an antigenic capsule, while gonorrhea does not. On a blood agar plate, if you were to streak this out, pictured on the bottom right, meningitis has a greater volume of growth while gonorrhea has a smaller volume. The key difference between both is though that gonorrhea infections have a high prevalence and low mortality, while Neisseria meningitidis infections have a low prevalence and high mortality. Outside of differences, meningitis has a DNA genome with a circular chromosome to hold its genetic information. It also has a plasmid to hold its antigenic properties, which is important in talking about its massive virulence factors. This bacteria produces through horizontal gene transfer, which means it replicates from the same generation and asexually, or how I like to learn is that they like to replicate wirelessly. They send their information over, and they have a new offspring, or not offspring, but rather they have a same generation to live on. In terms of their virulence, it will be explained in the prevalence slide next, since it is one of the most 3 common bacteria that cause meningitis. In prevalence, or the new and old cases combined, Neisseria meningitidis has a prevalence rate of 1.65 cases out of 100,000 population in developed countries. That means for every 1 person that gets infected in 1 week, another will get infected next week. The same idea goes if we escalate to developing countries around the world, and that means 300 out of 100,000 cases, meaning for every 150 people infected in 1 week, 150 more are infected in the next. As stated above, Neisseria shares its dominance with 2 other strands in Streptococcus pneumoniae, which holds for 50%, and Haemophilus influenzae, 18%. Neisseria rounds it out at 32%, and it primarily infects children aged 2 to 18, while the rest infect their own age group. Streptococcus pneumoniae infects adults, while Haemophilus influenzae affects infants. All 3 strands are prevalent during the winter and early parts of the spring outside of areas known as the meningitis belt. All of the prime strands listed above all have the same modes of action, but Neisseria brings a greater focus to the public because the only reservoir this bacteria can be found in is through humans, through their current infections or asymptomatic carriers, which will be explained later on. Now on to incidence. In terms of incidence or new cases only during a specific time, we can split this into 2 time frames: before vaccines and post-vaccines. The 1990s were before the advent of a meningococcal vaccine, and in this time, the incidence rate in the United States was 1.33 for 100,000, meaning that there were roughly 1.5 new cases of meningococcal meningitis in the United States on average per day. In the 2020s, where vaccines and vaccinations are in numbers and readily available, that number drops to 0.66 out of 100,000 population in the country, meaning less than 1 person was reporting to a hospital with meningococcus per day. In the same time frame worldwide, less than 1 person gets infected with meningococcus, and it lines up with the fact that vaccinations and vaccines are combating these numbers. The majority of the incidence rate today worldwide comes from the meningitis belt, which will be explained later on, which will be explained in the next slide, sorry. So, where can Neisseria meningitidis or meningococcus be a hotspot for infection? There are a few common sources ranging from places of travel to well-known hotspots. Places that are humid and places that are extremely cold, as explained in what time period this disease is normally found, in winter to early spring seasons. They are habitats for these disease. The most known place one can get infected by bacterial meningitis is through the contact with others in the meningitis belt of sub-Saharan Africa. I'll repeat that. The most common place one can get infected by bacterial meningitis is through contact with others in the meningitis belt of sub-Saharan Africa. There are places that can replicate what the meningitis belt is suffering through, and that is through unsanitary conditions, close contact with others, and unclean conditions such as constant windstorms, unbearable heat, unclean water, or just rather unclean conditions, like unsafe housing, things like that. In other places in the world, meningococcus is prevalent in the population due to it being part of the normal human flora. In up to 10% of the population, Neisseria meningitidis is carried asymptomatically with no harm to the host whatsoever in the nasopharyngeal region. It is similar to the behaviors of Staphylococcus aureus, where 10% to 30% of the population carried in their nasal cavity, which means they are both neighbors by coincidence. Another coincidence is that both are opportunistic. Once these bacteria find an open cut in the human body, they will enter and start their methods of propagation and infection. For those who carry meningococcus asymptomatically, it is extremely important to disclose it with your loved ones and those who reside with you, considering propagation begins with secretions and the handling of said secretions by another individual, so prophylaxis is a key component to intervention. As implied above, humans are the only reservoir of Neisseria meningitidis. In order, this bacteria is spread through secretions and close contact with infected and or asymptomatic individuals. This includes roommates and partners you are close with, example a romantic partner you share kisses with. Anything that involves close quarters with another individual or sharing of items like kitchen utensils leaves susceptibility to meningitis. Although Neisseria meningitidis is not like the more recent COVID-19, which spreads virulently through the air, it can still have another method of spreading in the form of inhalation of the droplets of infected individuals. Alongside what is stated prior in the idea that contact with the saliva of infected starts with the saliva of the infected starts the infection timeline. The effects of bacterial meningitis through Neisseria meningitidis starts with a puffy rash that quickly develops all over the body, followed by a stiff neck, fever, and headache. These effects are the preemptive conditions of bacterial meningitis, which can quickly escalate into a life-threatening situation if the person is either unvaccinated or delaying going to the hospital for treatment. The characteristics of Neisseria meningitidis include having a capsule, which is utilized by the bacteria to evade the phagocytes in the bloodstream and prevent phagocytosis by these latter cells. Once it finds a way into the respiratory tract or the meninges of the brain, which will be explained later on in the presentation, the pilus of the bacteria is used to adhere to the sites where it will multiply, feed, and further penetrate into the body. It ties into the fact that this bacteria is a chemoheterotrope, which means both its carbon and energy source are organic compounds, which in this case means the cells and the fluids of the body. Explaining the pilus having antigenic properties, it simply means two things. One is that the pilus carry unique protein codes that trigger an immune response in the body, making the host even sicker through a higher fever. The second item is that bacteria like Neisseria meningitidis has another property in its pilus that allow it to evade the immune system, and on top of that, allow for reinfection both with and without having an external source. This, on top of having a capsule, is why Neisseria meningitidis has a greater calling for attention despite being second for for infections of bacterial meningitis. It also adds to the cause with their size, making it even harder due to them being among the smallest virulent bacteria in microbiology. On top of that, this bacteria also employs an IgA protease, which means it has the ability to cleave antibodies of immunoglobulin A class, a signature ability of the Neisseria family. This is also the shared ability of other bacterial meningitis causative bacteria like Streptococcus pneumoniae and Haemophilus influenzae. It gets even worse. It gets even more virulent through the introduction of bacteria being catalase positive, and that means two things. The byproduct of breaking down hydrogen peroxide, which is water, oxygen, and water, it kills the immune response by lysing and bursting their cells. The usage of superoxide dismutase, which is an item that frees radicals of oxygen. It also lyses cells but immobilizes them to the point where catalase can easily break them down. With all this in mind, it makes a prime case for why Neisseria meningitidis is quickly lethal and brings about the most attention when it comes to bacterial meningitis. After getting into the organic compounds of the body, the Neisseria species can begin unleashing its most virulent actions up to this point. Using its endotoxins in the form of lipoligosaccharides, it can cause shock to the immune system and the normal flora of the human body, leading to their deaths through necrosis. At this point, the individual or actual host will be extremely sick and will be showing symptoms that include high fever and secretions, either through the mouth, the nose, or the bodily fluids. In other words, this is the exit portal of this disease. Since the cells inside, including the instigating Neisseria cells, have lysed, and they have the ability to self-lyse themselves, these secretions are contaminated and actively virulent for the next host to replicate in. This cycle begins again when the next person comes in contact with these contaminants. Building upon the idea that the Neisseria species draws the most attention, it is because there are two forms of infection timelines that this bacteria can follow. In the meningitis form, entry portals begin through the mucosa of the nasopharyngeal region and into the bloodstream. Similar to the signatures of Staphylococcus aureus, with it being part of the normal human flora for 10% of the population in the nasopharyngeal region, it also brings upon a coagulation cascade once it becomes an opportunistic pathogen. Neisseria brings about the circulatory collapse of the respiratory system and causes coagulation to the point of the entire system failing. After gaining nutrients from the respiratory system, this bacteria heads to a blood-brain barrier, where the meninges come into play. Hence its namesake for the virus. The meninges become inflamed, which causes the brain to swell. This pressure on the brain is called hydrocephalus, and before falling into a coma, the infected host experiences damages to their extremities or immediate loss of feeling in the central nervous system, which is the immediate brain and the immediate nervous system, sorry. So they will be losing feeling in their limbs, feeling in their legs. Immediate loss of feeling. It's not a good feeling. And also, before falling into a coma, they will be experiencing photophobia or sensitivity to light. And they will also be suffering altered physiological behavior. They will be very agitated, they will be very confused. Very severe symptoms will be falling upon them before falling into a coma. After a minimum of 3 hours without rapid or any treatment, the host falls into a coma and the secretions are expelled, and therefore you start the cycle all over again. The other infection form of the Neisseria species is through septicemia. Although you can quickly see and assume that both timelines are highly fatal, septicemia is different in that instead of a likely coma, death is more likely the end result through the symptoms to be explained. This form shares similarities through entering the bloodstream by the nasopharyngeal region and the lysing of cells causing this collapse of the circulatory system. But how septicemia is different from the meningitis route is that instead of only hydrocephalus and the passive consequences of this on the CNS happening, hemorrhaging is the end result of this route. Massive internal bleeding and pressure on the brain will cause seizures and then the trouble of controlling the rest of the individual, even if they are stabilized from the seizures. Alongside vomiting and diarrhea, death from septicemia can come along in sub-12 hours, making it one of the fastest causing bacteria for fatalities. I cannot show any pictures here, but I showed the only picture I can find where it doesn't show too much or just shows the least severe symptoms of septicemia, which is shown here. If Neisseria is quickly lethal, a public health response must be equally quick to counter it. But it shouldn't have to come to hospital intervention if the public follows the following actions in the presence of this bacteria. Prophylaxis is a good presentation to the public. Practicing good and proper hygiene is always at the forefront. Like with any virulent bacteria or virus, social distancing when being made aware that someone is infected or carries the virus asymptomatically, for example, is another prime example. Early diagnosis and vaccinations are the fail-safe methods of intervention, since species like Neisseria infects children ranging from extremely young to 18 years old. Identifying the disease in the infected can help lower the amount of time spent finding the right counter to the Neisseria species, which is important further on in this slide. In this day and age, vaccines are readily available, and even in the most viable incubation grounds, examples such as the meningitis belt or cold areas. Vaccinations are ranging from free to extremely affordable for those who need it or are susceptible to it. In the event of hospital treatments, the drugs that can be used ranges from penicillin and cephalosporins and cefotaxime, ceftriaxone, and vancomycin. The reason why I stated before that early diagnosis is needed is that there are subspecies of Neisseria meningitidis that are resistant to penicillin. Spending more amounts of time early on to find a proper counter if it is not penicillin is really a life or death moment in a short amount of time. If you're too late, the patient is in a coma or is unresponsive to the drugs due to CNS failure. In the next slide, we'll talk about the exact effect of these drugs. Depending on the strand of Neisseria meningitidis, it is susceptible to penicillin. Since Neisseria is gram-negative, it is highly drug-resistant, so it makes sense to wrap up all these slides prior in the quick and early intervention is the key. In the fact that quick and early intervention is the key to combat this bacteria. The effect of penicillin is that it breaks down the cell wall and allows for the cells to lyse early on in its life cycle. The patient will still get sick due to its endotoxins, but never to the extent if Neisseria truly propagated in the body. In the event that the patient reports with a penicillin-resistant subspecies of Neisseria, any combination or single usage of the cephalosporin from cefotaxime, ceftriaxone, and vancomycin are the most effective treatment plan. These drugs attack the protein synthesis of the bacteria, killing it before it can attach or release its antigenic properties on the host or the immune system. In the case that the infection propagates to the meninges or the blood-brain barrier, the usage of these drugs can quell or even stop hydrocephalus from being the fatal result of the patient. In a normal Neisseria strain, penicillin can be combined with any of the cephalosporins to form a symbiotic effect in the mode of action. A prime example is that penicillin can break down the cell wall, so cephalosporins can have a straight path to destroy protein synthesis in the bacteria. And that wraps up my presentation. I really hope you guys enjoyed my video, and please follow for more.